So while the slides are coming, I'll just mention, so today I'm going to talk about a new project called HippoMaps. So basically here, we're using HipUnfold to gain like surface fitting to many different kinds of data set and then aggregate them together in order to look at combinations of different modalities and try and use that to leverage some integrative information. So here we are, HippoMaps, multiscale cartography of the human hippocampal organization. So this is very much inspired by NeuroMaps, a project by Bratislav Misic and his lab. So basically what they're doing here is mapping out many different features to the neocortical mantle, including MRI features, intracranial EEG features, histology features, many things. And what they do with this information is, well, many things. So one thing you can do is take one individual map and then compare it to other maps in order to gain some profiling, if you like, of that individual map or that individual person or patient population, if you like. Another really useful thing is that you can take these maps and look at differences between them or similarities between them in order to sort of contextualize what they may mean, or for example, what a new kind of signal acquisition might be telling us about the brain relative to other maps. Of course, this is all done in the neocortex, where we have this very nice alignment system. So the neocortex, of course, has different gyral and sulcal patterning, which we try to overcome generally by inflating this structure to a sphere and then registering these spheres between each other. And then we can apply an atlas to that sphere or to a folded brain, and then we can take that sphere and project it back into any one subject's space. So in a way, this is trying to overcome problems with inter-individual variability. And as you guys know, I've been working on this problem in the hippocampus for a long time, where basically the hippocampus is cortical, as Boris mentioned, so it's actually archicortex or old cortex, and so it has this folded shape as well. And we've been unfolding it to a rectangle. And the reason that we do this is basically to align different individuals with different morphology. So on the left here, I have one hippocampus that has a given folding pattern, and on the right here is another sample actually from BigBrain, which has a very different shape and a very different folding pattern, including a different number of gyri and sulci along its excuse me, anterior and posterior axis as well. So we have software to do this. I think some of you are already using it, and I won't go into the details of it, but you can talk to me about it if you're curious. And I'll also just mention, if you guys remember last year, we also published this paper refining the registration between individuals by basically doing 2D registration in this unfolded space. And we found that we could get down to about 0.5 millimeter alignment with that kind of method. So we're not aligning cells or things like that at the very micro scale, but this is kind of the order of magnitude that we're aligning individuals on. So as I kind of mentioned at the very beginning, this HippoMaps project, basically what we want to do is create a repository of different maps coming from very different kinds of data, and then use it to look at combinations of that data. So the basic approach is we want to be scale invariant, I suppose. So at the top here, I'm just showing some slices of BigBrain with a surface fit to it, and then you can see the folded and unfolded surface next to it with a very high density or a very high number of vertices. And then on the bottom, we have a more standard sort of 3D MRI-like image where we have less detail, less folding, but we can still map it to the same unfolded rectangle with a smaller number of vertices. And in that unfolded rectangle or unfolded space, we can do all kinds of operations. So we can map out features of that surface itself, for example, its thickness or its curvature and things like that. And then with those features, we can actually use that to drive a registration between individuals, or we can do things like parcellation. So we have sort of a canonical unfolded subfield atlas, which comes from BigBrain and several other histology data sets as well. And we can also do operations. For example, we can average between subjects, or we can run spatial correlation between subjects, or all kinds of things, all kinds of statistics in that standardized sort of unfolded space. So I'm going to take the parcellation atlas and just put it off to the side to keep as a reference for how this unfolded space is sort of organized. Maybe I should also mention the anterior is at the top, the posterior is at the bottom, the lateral is on the left, and the medial is on the right. So within HippoMaps, we've then mapped out many different kinds of data. So I'm just showing as an example here from the data set Boris mentioned, looking at 7T MRI. Here we're looking at a fairly universal contrast, quantitative T1. And you can see in this unfolded space, it actually differs in line with its subfields. So this sort of light blue color, it's CA1, and it shows the highest intracortical myelin, or quantitative T1. So this is one example. We have many different data sets that we actually looked at with this approach, and I'm not going to show every single one because I think that would be exhaustive, but I'll just highlight a few. Here's another one from the same 7T data set where we did diffusion MRI and extracted the feature of apparent diffusivity. And so this gives us an idea of how much water is moving within that tissue, so it gives us some idea of the tissue properties. And again, we can map that to this unfolded hippocampus, and now we have a different map that's highlighting different subfields and differentiating different subfields. We can also do things like add cortical depth to this data as well. So here, this is the polarized light imaging data set that actually Alexander Oberstrass also worked on and Timo presented yesterday. Here we're just looking at transmittance, so basically how much light is passing through this tissue. And we did this across multiple surface depths. And so what we can do from that then is get a profile, as Boris was mentioning, these like microstructural profiles from each vertex. And then we do some post-processing to bring them into even closer alignment. And then we can do things like look at some individual profiles coming from, in this case, coming from the distal side. So this would be more towards like the CA3, CA4, and dentate gyrus, versus the proximal side, which would be more like the speculum and becomes a bit more cortex-like. We start to see a more bimodal distribution of neurons. Yeah, this just, I think, highlights some of the advanced options for HippoMaps. We can also look at functional MRI. So here it's just an fMRI time series projected onto an unfolded left and right hippocampus. And then we can, as Boris also mentioned, look at correlations of that time series with neocortical parcels or vertices as well. And from that, we can then take the correlations between hippocampal and neocortical vertices. And if we average over the neocortical vertices, we can see which parts of the hippocampus are on average connected to parts of the neocortex, which is what I'm showing in this unfolded map. And the output structure here is, the highlighted structure is on the right, so it's the subiculum, and this is actually the primary output of the hippocampus, so this makes a lot of sense neuroanatomically. And then at the bottom, we can also average over the hippocampal vertices and see which parts of the neocortex are connected to the hippocampus. And as Boris mentioned as well, it seems to be a lot of default mode areas. Yeah, so just describing the data set this comes from. Some of the other cool things you can do is you don't actually necessarily need to use image data. So you could actually use scattered data. For example, what you would get from intracranial EEG recordings, where you just have a few contacts placed somewhere in the hippocampus. And then we use a registered MRI image to derive a subject-specific hippocampal surface. And then we look at where those contacts intersect the hippocampal surface. So I'm just showing two example channels. And then in this case, we use some extrapolation and interpolation based on the distance from where those contacts are across the hippocampus. And from the intracranial EEG time series, we can extract information like power spectrum density and also the band power of specific bands, like the alpha and theta bands in the hippocampus. And then we can also then, across many channels, so 81 here in the hippocampus, we can then generate a map of what parts of the hippocampus show, in this case, stronger theta power, which here we see more towards the anterior and more towards the medial side. Yeah, those were a few examples of the new maps that we've derived. I also just wanted to mention that as part of this project, we want to encourage community involvement. So this is what I would call a community contribution. So a colleague at Université de Marseille, Roy Haas here, did this awesome study looking at 7T arteriospin labeling and perfusion of blood through the vessels surrounding the hippocampus, which you see in the top figure. And then in these bottom two figures, he actually mapped out features of those vessels into this standardized unfolded space. And so within this HippoMaps data repository, he has then uploaded that data in a relatively lightweight sort of GIFTy format. So it's much lighter than the original like raw data or even the pre-processed data. It's just a small number of vertices here. And so we can then take that map and compare it to our existing maps. So, for example, Alexander Oberstrass and his PLI features, we're also uploading those features. I won't get to talk to it today, but we're also sort of searching for more features. So, for example, we're pulling data now from the Allen Mouse Atlas and also some other projects that some of you guys are involved in. So I won't get into all of them just because of time, unfortunately. But I want to focus on one more thing, which is once we have this huge collection of maps, what can we do with them? So one of the things that I think is most interesting to do is to compare them via spatial correlation, which we can make sure is fairly robust by some permutation testing. So here I'm just showing a collection of all of the maps that we included in this initial study. So at the top we basically have, in this like sort of cyan band here, we have histology features, then we have MRI features, then we have fMRI features, then we have intracranial EEG features, and the intrinsic features of the surfaces themselves, so like their curvature and things like that. And you can see that within each of these modalities, there is a high correlation between the modality, but also some interesting correlations across modalities. So for example, correlations between histological features and MRI-derived features. So this, I think, is one way that we can explore what is it that our MRI contrast is actually telling us about the tissue itself. As Boris was mentioning, we can also use the same sort of gradient mapping or gradient decomposition technique. So here, I actually used as the input features all of the different maps that we had derived in this like large multiscale multimodal data set. And then we get a few gradients that explain variance in that data. So the first one here basically goes left-right or proximal to distal. And this is basically differentiating in the same axis as the subfields of the hippocampus. The second one here goes a bit more anterior to posterior. You can see it especially in the sort of folded shape of the hippocampus. So true anterior to true posterior, with also a little bit of differentiation between subfields as well. So just exploring that a little bit more, here I'm plotting all of the vertices of the hippocampus. The top map then is coloring those vertices. So, sorry, I should say the x-axis is the first gradient, the y-axis is the second gradient value. And then each vertex is colored according to its subfield in the first case, according to a continuous proximal to distal gradient in the second case, or colored according to its anterior-posterior location in the third case. And we can see that they're actually, they get sorted fairly well into these different axes, just based on the data alone, which is not aware of the closeness of those vertices. So I'll focus mostly just on the subfields and the anterior-posterior axis. What we can then do is actually take all of our different kinds of features, and we can map out whether they're correlated with, on the x-axis here, the anterior-posterior axis of the subfield, or sorry, of the hippocampus. So here again, I'm just showing different colors representing different modalities. So we have features like intracranial EEG in purple there, which were generally quite correlated with this anterior-posterior axis. And then on the y-axis here, I'm showing the different subfields as discrete labels. So we're doing a Pearson correlation here. And you can see features like a lot of the structural and histological features were highly correlated with the subfield axis of the hippocampus. So I think this is really useful because we can then use it to take new information and contextualize it with respect to subfields, the anterior-posterior axis, and also potentially with all of the other maps that we've collected in this repository. So I'm just going to show a quick example of what I mean by that. So let's say, for example, you're a psychologist and you run this study test paradigm where basically you're testing people's memory, and you do it in an fMRI scanner. And so then you can map out the hemodynamic response function for different memory performances. So to totally novel trials, or to trials where people were able to do pattern separation, so basically maintaining a very good representation in memory. And we get these maps then of which parts of the hippocampus are most involved for each of those kinds of memory. And then we can map out, for example, in this novelty map, we can see that it's moderately correlated with this anterior-posterior axis of the hippocampus, suggesting that anterior to posterior is maybe a good way to understand the functional specialization of the hippocampus. We can see it's also moderately correlated with these resting state fMRI and intracranial EEG features. Then we can also take this other condition, so pattern separation, and we can see it's actually more correlated with the different subfields of the hippocampus. So it's likely driven by a different sort of subfield microstructural feature. And again, we can compare it to the other maps in that feature as well. So altogether, we presented here 30 or more, depending on how you count them, new maps that span things like histology, structural and fMRI, intracranial EEG, with sort of open, like community contributions as well from all kinds of different data sets. Importantly, this is meant to be flexible to different kinds of data, so not only different resolutions of imaging, but also things like scattered data, such as intracranial EEG or even tissue punches that you can then do genotyping on and different things like that. And again, the final maps are relatively lightweight, so we've provided a lot of Python tools where you can just quickly download and compare these maps to your own maps as well, and hopefully learn a lot of information about what your map means. Within some of the maps that we looked at here, we saw a lot of separation of the different subfields, especially with microstructural sensitive features, so things like histology, and then also this anterior-posterior differentiation, especially with what I would think of as sort of macro-scale connectivity, such as its connectivity to the rest of the brain via fMRI, or band powers via intracranial EEG. So the overall goal is, new people, new users now can either upload their maps or download these maps and learn something about their own data as well. So the tool is here. It's, as Boris mentioned, under review at Nature Methods. And then also, as Boris, I'll thank everybody who contributed to this, which was many people, and I appreciate it very much. I also just wanted to mention really quickly, for the AI enthusiasts in the crowd, this perspective paper that we have just preprinted that basically tries to use this organizational principle of the hippocampus to inform how we might design AI. And especially this notion that the hippocampus may be a supervision signal for training the rest of the neocortex. With that, I'll wrap it up, and I'm happy to take any questions.